Hi folks, my name is Cole, and today on Investigate, Explore, Discover, we're going to be looking at neutrophil function in SARS-CoV-2 pathology. So stick with me through this whole video to get all of the background information so we can dive into some exciting experimental results. First up though, today we're talking about SARS-CoV-2, and SARS-CoV-2 is the causative agent in COVID-19. This virus was first identified in China in December of 2019, and has since then spread all over the world. And the World Health Organization declared it a pandemic in March of 2020. Since we first discovered it, daily new confirmed cases of COVID-19 have continued to rise. And they haven't been rising equally throughout the world. There are multiple places around the world that are just hotspots for infection and have had more cumulative cases than others. Worldwide, there's been a cumulative confirmed caseload of over 157 million. And has been a contributor in over 3.2 million deaths. Now, SARS-CoV-2 is a coronavirus that has a double-stranded RNA viral genome. Of particular note is one of the proteins on the outside of the virus called its spike protein, because this protein allows it to interact with host cells. In particular, it interacts with a couple different receptors on the outside of host cells. It interacts with ACE2 to mediate attachment, and it interacts with the serine protease TMPRSS2 to mediate its activation. And so once it's attached and activated, this allows the virus to enter into host cells and start replicating. And once the virus starts replicating to high enough levels, we start to exhibit symptoms. Now, upon infection with viruses, it's difficult to tell by symptoms alone what we are infected with, because our body has many generalized responses to respiratory viruses. So symptoms of allergies, symptoms of being infected with the common cold or influenza, all can look similar to being infected with COVID. This is why it's important to be able to identify disease profiles to tell us the differences between common infections and can lower our general apprehension, which I found was very high at the beginning of the pandemic, where you're giving everyone side eyes if they so much as sneeze. COVID-19 typically presents as a cough and fever, and most characteristically a loss of taste or smell. So as you can see though, there are many different possible symptoms you can have with COVID. And alongside many different symptoms, there's also a diverse range of clinical manifestations. Most people will experience mild to moderate disease. However, there are some that experience severe to critical. And at this point is when COVID-19 is commonly associated with death. Now, this increase in disease manifestation is typically brought about due to COVID-19 causing an increase in inflammatory signals, which overactivates our immune system to cause something called a cytokine storm. And cytokine storms are not specific to COVID-19. Cytokine storms can happen from many different infections, like sepsis, for example. Cytokine storm can lead to many severe outcomes, such as acute respiratory distress syndrome, thrombogenesis, multi-organ failure, hyperinflammation syndrome, shock, and death. Now, our immune systems do mediate this, but our immune systems are made up of a plethora of cells. The cell that we're going to focus on today are neutrophils. Neutrophils are part of our innate immune system and have a generalized, fast-acting response to pathogens. Neutrophils are a very abundant cell type. And in any immune sampling, neutrophils can make up anywhere from 40 to 70% of the immune cells in a given population, and are some of the first cells to respond to infections. Neutrophils are able to interact with their environment through a variety of different receptors that they have on the outside of their cell. This mediates their effector function. One of the most unique ways that neutrophils function, though, is through the generation of neutrophil extracellular traps. Neutrophil extracellular traps, or NETs, are extracellular fibers composed of DNA with histones and granule-derived enzymes such as myeloperoxidase and elastase. NETs have been shown to have antibacterial properties. However, this can be a double-edged sword and have been shown to function in tissue injury. In fact, increased neutrophils and NETs are implicated in the pathogenesis of several diseases, such as rheumatoid arthritis, diabetes, and sepsis. This brings us to why it's important to look at neutrophils, particularly in the case of COVID-19. This is because neutrophilia, or the increase in neutrophils, and cytokine storms are present in COVID-19 and other diseases. So by determining the mechanism of action during COVID, we can ascertain what type of drugs may be utilized for effective treatment. This brings us to the specific paper that we're talking about today, which is SARS-CoV-2 triggered neutrophil extracellular traps mediate COVID-19 pathology by Veras et al. From the University of Sao Paulo, Brazil. And in this paper, they investigate cellular and molecular mechanisms underlying NET production and their immunopathological role in COVID-19. They do this by comparing healthy patients to COVID-19 patients. And one of the first things that they compared was the level of neutrophils in their blood. They found that COVID-19 patients had high levels of neutrophils and also high levels of neutrophil extracellular traps, or NETs. When isolating these neutrophils, they also found that the neutrophils from COVID-19 patients were able to be stimulated more easily and more readily produced neutrophil extracellular traps. So COVID-19 patients produce high concentration of NETs. Now, I want to take a moment and just draw your attention over here to what neutrophil extracellular traps even look like, or really what is going on at the microscopic level in our body, because we don't get very many chances to actually see what's happening. So just take a moment and admire it, because damn, that's a cool looking picture. In addition to looking in the blood, they also looked in the tracheal aspirate of people affected with severe COVID-19, and they found that in this location, there was also an increase in neutrophil extracellular traps. And again, you can see it right there. You can see what these cells are doing. Like, oh, it's so cool. You don't get to see that so often. And in patients that deceased due to COVID-19, they took a look in their lungs. And again, they found an increase in neutrophil extracellular traps. So this prompted these researchers to ask, how are NETs being produced? And they investigated this by taking healthy patient neutrophils and co-culturing them with SARS-CoV-2. At which point, they looked closely at the neutrophils and imaged what was happening. So as some baseline controls, what they first did is looked at a mock infection and looked at neutrophils infected with inactivated SARS-CoV-2 virus. And as you can see, there is no NETs present at all. Now, when they infected neutrophils with increasing viral loads, they saw that as the virus load increased, the amount of NETs were also increasing. And furthermore, when they inhibited the production of NETs, no NETs were seen. So it's a good control to include. They also found that in neutrophils that were undergoing netosis, or the expulsion and creation of neutrophil extracellular traps, there was more SARS-CoV-2 present around. They also saw that viral replication continued as time progressed. Now, the next question that they asked is, what exactly is facilitating this netosis? As we know, neutrophils have many receptors on the outside of the cell. So they looked at how SARS-CoV-2 was interacting with them. They first postulated that, it's interacting based on the normal mechanisms through ACE2 and GMPRSS2. And when that happens, you have netosis, the expulsion of NETs. But when blocking ACE2 with antibodies and disabling the serine protease activity from TMPRSS2 and co-culturing with SARS-CoV-2, there was no NETs produced. So these receptors are critical for SARS-CoV-2 function in neutrophils. Now, because COVID primarily affects people's lungs, they next asked, what effect does netosis have on lung epithelial cells? So what they did is they took SARS-CoV-2 activated neutrophils and co-cultured them with lung epithelial cells. And in doing so, they found that these lung epithelial cells were undergoing apoptosis and dying. However, when they degraded NETs in cell culture, this allowed the epithelial cells to survive, indicating that these NETs are what is causing epithelial cell death and damage. So, altogether, what they found is that SARS-CoV-2 interacts with neutrophils via their ACE2 and TMPRSS2 receptors to cause netosis and the expulsion of neutrophil extracellular traps, which causes these epithelial cells to die and furthers COVID pathology. This knowledge, number one, is interesting to learn about, but significantly increases our knowledge of what is occurring during SARS-CoV-2 infections. It does this by showing that neutrophil extracellular traps drive worsening severity of COVID-19 and identifies how active SARS-CoV-2 is required for netosis, because if you recall, inactivated viruses do not cause netosis. This indicates that NET inhibitors may be an effective treatment for severe COVID-19 patients. Now, all science is basically just a step forward in our gathering of knowledge. And to continue making those steps forward, we have to be asking more questions. And there were a few questions that kind of stuck out to me when going through this information. The first question is SARS-CoV-2 replication required for netosis? Because inactivated virus do not trigger this. Furthermore, once inside of the cells, regardless of what's happening, what receptors in the cell are recognizing SARS-CoV-2 and thus inducing netosis? These are important things to know so that way we can get a better cellular understanding of what is going on. And finally, because there are many different symptoms in COVID, how else are NETs driving disease? Are NETs even important for these other symptoms? Or are they only really important in the lungs? Love to learn more about it. My final question, though, is what sort of ideas or questions popped into your head when hearing about this information? Let me know about them in the comment section below. And also, let me know if you have any topics that you'd like to hear about more in the future. Ultimately, though, I hope that you learned something, and more importantly, you enjoyed your time doing so. So if you did, give this video a like and subscribe for more in the future. And with that, I hope you have a great day, and I will see you next time.